Hi everyone, my name is Brian Schilder. I'm currently a PhD candidate at Imperial College London, but today I'm going to be presenting some work that I did as a bioinformatician in the lab of Tapiq Raj, along with Jack Humphrey at Mount Sinai Hospital. When one conducts a GWAS or a QTL study, we end up with a set of regions in the genome which contain variants associated with the given disease or phenotype. However, non-independence between variants due to linkage disequilibrium makes it very difficult to distinguish between the causal variant or variants underlying the disease and variants that are simply highly correlated with the causal variants. Fine mapping is a set of statistical techniques designed to identify the most likely causal SNPs. In this way, we can think of GWAS QTL studies as the paper roadmap that gets us to the right city, whereas fine mapping is the high precision GPS that gets us to the specific house we're trying to find. However, fine mapping is often underutilized largely due to the technical difficulty of setting up idiosyncratic input-output formats across different programming languages, gathering the necessary data to run these tools, such as LD panels or genome-wide functional annotations. Also, each fine mapping tool has its own set of strengths and weaknesses, which can produce different but often overlapping credible sets of variants with 95% probability of being causal for the phenotype. We therefore developed an R package called Echolocator to integrate many of the most commonly used tools and datasets to seamlessly perform fine mapping across all loci. All that is required by the user is a path to the full GWAS summary statistics file and certain column names. This offers several main advantages. First, it enables a high degree of reproducibility. The full pipeline can be run in a single R function, which fine maps all loci with multiple tools, generates locus plots with multiple window sizes, conducts functional enrichment using a wide range of data sets, and organizes the results in a standardized hierarchical folder structure that facilitates further downstream analyses. Second, it's robust in that it enables the identification of several high-confidence consensus SNPs per locus, nominated in the credible sets of multiple fine mapping tools. This also has the benefit of drastically reducing the number of putative causal SNPs from an average of 85 genome-wide significant SNPs per locus to just 3 consensus SNPs in our recent study of Parkinson's disease. This is ideal for nominating SNPs for experimental validation, such as CRISPR editing in relevant cell types. Third, its fully integrative nature permits very rapid, comprehensive fine mapping studies. Thus far, we have used it to fine map 11 different neurological disease GWAS, which enabled our recent investigation of shared causal genetic variants across these diseases. Lastly, we provided API access to hundreds of functional annotations, which includes cell type specific epigenomics, as well as predictions of functional impact probability from a suite of machine learning models. As an example, I'm going to briefly go through some of the main outputs of Echolocator in our recent application of this tool to the latest Parkinson's disease GWAS by Knowles et al. Here we see 2 locus plots generated by Echolocator side by side, with the negative log of the GWAS p-values shown at the top row. In the rows below that, you can see that the various fine mapping tools propose different credible sets. Only some of these are agreed upon by multiple tools, and these are defined as the consensus SNPs. In the bottom half of the same plot, you can see the consensus SNPs, indicated by gold vertical lines, fall within enhancers that are exclusively active in oligodendrocytes in the DYRK1A locus on the left, and neurons in the FCGR2A locus on the right. Another tool integrated into Echolocator called Motif Breaker queries a large compendium of position weight matrices to identify the transcription factor binding motif that these consensus SNPs fall within, and to what extent they disrupt their binding. Across loci, when looking for overlap between consensus SNPs and cell type specific epigenomic peaks, and interactome anchors on the x-axis here, You can see that each locus has a cell type fingerprint, sometimes impacting multiple cell types, other times primarily impacting only a single cell type. This can help us to not only dissect genetic and cellular architecture of a given disease as a whole, but like Parkinson's disease, but also to make predictions about the primary mechanisms at play within a given individual's particular form of the disease in an effort to move to a more personalized approach to disease diagnosis and treatment. Lastly, I'd like to thank all of my collaborators and funding agencies and direct you to check out some of our recent preprints describing this work in more detail, all of which are currently available on bioRxiv. Thank you.